Uh, First, it was about what you can do in order to address red tape. And one very clear thing of red tape we have in Europe that we don't have in the other areas is that you are forced, in addition to get the clinical trial approved, you also need to go to another authority to get an EMO approval for these kind of products, which is a kind of red tape that has been developed for seeds where there is a risk that it could replicate in the nature. It's no such risk for the pharmaceutical product because outside the body not be working freely. So that's one of the things that complicates making clinical trials in Europe. There is also some commercial issues that you need to adapt very stiff systems to get pricing and reimbursement approved that are not dynamic. And my colleague from the European Association of Hospital Pharmacy was alluding to having a more flexible way of paying for this kind of products. For example, pay if you got an effect, for example, and if you do not get an effect, you shouldn't pay, just as one example. And then there is a general issue within the European medicines regulatory network, because actually assessment comes from the member states. And that is that there is a lack of resources, and I believe even if you do some sort of efficiencies, I still think that you need more resources, both in the form of expertise and also in the form of money to finance this. Finally, you were asking about ICMA, the International Coalition of Medicines Regulatory Authorities. These consist of the heads of the medicines agencies, and I think that they have an important role to set the standard and the framework what other organization initiatives should do, and thinking about the International Council on Harmonization, which I think could give a good framework for gene and cell therapy. So far, the leading authorities have thought that it's not mature enough to develop this kind of guidelines, but I know that our sister organization BIO will come up with a proposal pretty soon, again and raise it to have a kind of reflection paper to start with, and then continue with guidance, and then you will address one of the main issues that you have with different rules and regulation, or lack of rules and regulation and guidance that you have in several markets.
Just wanted to add, when it comes to Europe being the slopes of the three regions, that doesn't really have to do with EMA. It has to do with clinical trials which are being assessed and approved at the national level. There is a lot of variability and bureaucracy. And because of this reason, the European Commission a few years back, they wrote the clinical trial regulations, which came into effect last year, that from this January it will be compulsory for any new clinical trial. And then it will be one application, one decision for whichever countries you want to run the clinical trial. And hopefully this will streamline things a little bit. And going back to global assessment and approval of the drug, that's still a long way off, as Professor Kramer said, but there are already a few steps. I mean, you can think of EMA, we're already a step there. You know, you have a pan-European approval. But also the past few years there was a consortium that came to being, which was between Australia, Canada, Switzerland, and Singapore. You would make an application, and those 4 agencies, they would assess your application together. They would still take a decision of whether they get you approved or not individually, but that's already a first step. And since Brexit, UK has also joined that consortium, which is called the Access Consortium. So it takes a long time, but some steps have been happening.
Okay, I saw there is one more question over there.
So my question is in regards to different companies having different results in genetic predictions. Would it help to have a single genetic library so that statistics is the same and they can give results based on that?
Would it help? Certainly. Which company would be willing to adopt another company's algorithm? No one. It's what they call the USP, the unique selling proposition. Every company thinks that they have the best algorithm. And apparently, if you have result A from company A and result B from company B will say that the result from company A is wrong. Okay? It was also an issue of which platform you're using. If you're using a specific genetic screening platform or another. For example, a different company. There are a few companies that they have similar platforms. So this is the basis of their explanation of the different result. So everybody is happy in terms of the company. The patient is not. And that's the bottom line. And of course the advisor is very happy because he gets money. We had a very well-known geneticist here in Athens that got almost 10,000 euros per month just to provide advice and to expand on the knowledge of PRS. And then the whole thing fell flat after 2 months when he decided to just move. Okay? Actually, both ways were unprofessional. So we should be very prudent as scientists. We should be really aware of what is the power we have at hand. It shouldn't be abused. It should be properly exploited to improve the quality of life of the patients. And we should be very, should be, I think, skeptical, critical to what we hear. And pharmacists are going to be asked to promote these specific tests because some of those are provided over the counter. And you have to be also very vigilant. Try not to follow the quick profit, the quick bucks. Try to be very prudent with how you advise your patients to get the proper solutions for them, health-wise.
Okay, so maybe we'll. Okay, I see we have some more questions from the audience.
I have a question about the last presentation. So you said that in the meta-analysis you exposed 38 non-associative genes that companies use to supposedly personalize the nutrition or drug regimen. I was curious, is there any authority to take down those companies after, because it is an unethical claim to claim that some of the therapies could improve patients' health even though they don't, and clearly it's very expensive. So is there an authority to control such claims, or do they have a right to do that because the legislation hasn't caught up? Thank you.
Good questions. I think you were following. So the answer is simple: no. The problem is that those particular companies are genetic analysis companies. They're not medical companies. So that particular company that does the analysis for infidelity tests, they also test stallions for the genetic homogeneity stallions, the horses. Okay, yeah. No, I use it actually verbally, not metaphorically. So there's no legislation, unfortunately. This study was just to bring up to light that a particular genetic analysis laboratory in Athens, that also had subsidiaries in other cities, were actually abusing this information and actually didn't know what they're doing. And that's why we focus on those particular 38 genes. There are numerous genes that have been associated with responding to that with nutrient intake, and what is worse, nutrient-related pathologies. That's even worse, diabetes, obesity, and the problem is that this particular company had the nerve to go to primary schools and try to advertise their services, while they have been specifically instructed not to do so by the ministry. So they're also using unfair means of advertisement. They have no ethos, right? But what society does about it? Nothing. You don't have the tools. So we have to try. And if we fail, we have to try harder. Okay? We fall because we learn how to stand back again. So that's the idea. And we have to keep trying. It's the only way that those companies can go out of business. They went out of business. There are other companies that rose, and they will be brought back again. That's easy. That's for sure.
The only thing I would add is many of those devices that you see and tools, they got approved under the Medical Devices Directive. There is the new medical devices regulations that is coming. And for many of those companies, they will find it very hard to keep their product on the market, or even to bring new products in, because the hurdle to get approval will be a lot higher because of these new regulations. So I don't think it will stop it, but maybe it will make it a little bit harder.
Thank you a lot. And now we will come back to Slido. And the question is, what are hurdles we need to overcome in order to lower the costs for gene therapy and make it more affordable for everyone?
It's a difficult question to answer, but I think, at least my understanding is that you have shown the great value of this kind of new therapy compared to what the alternative could be, lifelong treatment of a certain kind of disease, and it could be costly operations that you need to do as the alternative, or the alternative could be also that you die if there is no other things. So I think from a health technology point of view, I think it's possible to show that these very high costs are relevant from the kind of algorithm that you have on health technology assessment. But then, as I said before, I think you need to find either way of paying for this, like as I said, that you should only pay if you get this result that you hope for, and you could also consider to divide the payment over the years to continue the follow-up and see that it's still working, because you do not have 100% certainty that it will be working for very many years. And so I think you need to find more flexible way of paying for this kind of therapy. So that's what I could say. And then from a general point of view, the more these kind of products you will get competition, and then also it could have an effect, slight effect on the prices. I don't know if any of my colleagues want to add.
I think one thing that is adding to the cost of all these innovative products is the manufacturing and supply. So it's one thing a pharmaceutical company saying this is what it costs to make it and supply that on this fine. But if you have, especially smaller countries like Greece, you've gone through an economic crisis, you go through COVID, there's not a lot of budget there for all the medicines that are needed. So very often it's also, I think, the responsibility of ourselves, the industry, to collaborate locally and with other organizations. How can we bring the manufacturing costs down, and how can we make sure supply costs are also down? So it's a holistic approach that both industry and national level government organizations can need to take.
I would like to ask you, are there any other questions? Because before I saw some hands raised up. So are there still any questions from the audience?
Okay, thank you. This is more for regulatory affairs, I guess. In terms of omics and therefore advanced therapies, it's kind of hard for the regulations to be up to date and be just to keep up with the new innovation and everything. So I would like to hear from corporate and FB's point of view, how do you deal with these hurdles and what do you do to try to change it and make it a more sustainable environment for these new therapies? Thank you.
So one way to approach this is speak to the regulators early. There are already certain regulatory pathways at both FDA and EMA level that you can get your product designated if it's innovative, if it has the promise of providing significant benefit to patients. Once you're designated through those pathways, then you have a lot more frequent interactions with the regulators. You can discuss what my phase 1 study needs to be like, what about my preclinical package, my manufacturing capabilities, and how can we get to the market much quicker? I think as Professor Teller mentioned, many of those drugs, they will be orphan drugs. What this means is that it's actually for rare diseases, for smaller populations. And again, there are regulatory pathways that you can get into the market quicker or use some innovative statistics, for example, if you have very small number of vaccines. And as I said earlier, regulators themselves, they also learn. They don't have all the answers. So the more we meet with them, especially in the innovative program, the more we share, and then the smoother the pathway can be to the market. And then the other aspect is regulatory policy. As we have more and more of those products coming through in industry consortia and fora, we discuss those compounds, we discuss where the gaps are in the legislation, and then we try to influence regulatory policy and regulations and guidelines so that they start accounting for those innovative products.
I have nothing further to add. This was very comprehensive and good. Awesome.
One important aspect that refers to implementing innovative personalized therapies is guidance heterogeneity. So if I wear now my regulatory hat, being at the EMA as a national representative of Greece for 12 years, I can tell you that one major issue that we have is that FDA says different things than what EMA says. And it's a huge problem because it does not refer to what they say, but also how many drug gene pairs are referring to. So at the EMA, we have something like 140 drug gene pairs, and the EMA has over 300. And also the level of importance of using this particular guidance is different. So FDA may say that's testing recommended, while EMA says that's just for information, and the other way around. So this really it creates confusion to the clinicians that they will be using this particular knowledge. And also, despite the fact that we have bilateral and trilateral goals between the FDA, the EMA, and the PMDA, that's a Japanese agency, still there is a huge discrepancy between the guidance, not the guidelines, the guidance that provided by those regulatory bodies. If you put on top of that the differences that exist between research consortia like the Clinical Pharmacogenetic Implementation Consortium or the Dutch Pharmacogenomic Working Group, then it makes the problem even worse. So for that, we need to establish a consensus. It's a huge undertaking, but it's something that's really needed because the clinicians understand a specific guidance and need to follow. So if they're in the US, they probably follow different guidance compared to the European clinicians. Not to mention what happens in countries like Middle East, Asia, and Africa.
Thank you for your answers. We will proceed to the next question from the audience. Are there any questions?
So as you mentioned that there's nothing that is being done about genetics, and my question is what could be done in general and for us as a pharmacy specialist to assure or at least somehow improve genetics?
So yeah, the only way is awareness, education, is what we're doing here. It's beautiful weather outside, but still you're here in the room trying to absorb some of this new knowledge that we were trying to get across. It's an endless effort. You're going to get to learn new information about emerging genetic technologies, be it individualized treatment or gene therapies. You should see everything critically. And critical means that you can understand whether a piece of information is provided objectively in an academic fashion or provided as a marketing approach from certain companies. So I'm certain that you are very, very, in fact, clever to understand this particular difference. So the only advice to you is think critically and act objectively for the benefit of your patients. That's the only way forward. And believe me, in the medium to long term, this is going to be appreciated. In the short term, some of those companies may make money. It won't last. I can tell you. It's just a balloon. It's going to explode.
Thank you. So we'll proceed to the next question, this time from Slido. The question is, seeing as siblings and twins share basically the same DNA code, is it fair for only one of them to decide when to submit the data to companies like 23andMe?
I heard the first part of the question. Two siblings?
Seeing as siblings and twins share basically the same DNA code, is it fair for only one of them to decide when to submit the data to companies like 23andMe?
That's a question. Monozygotic twin or dizygotic twin? So dizygotic twin is basically different. If it is a monozygotic twin, then the issue is environment. Basically different and different. So it's not just genetics, it's not just genetic fate, it's not our genetic predisposition. We're more than genetics. Okay? So even if one monozygotic twin sends his DNA or her DNA to the company, and then you have another one that gets identical information, it's not certain that he or she is going to develop, and she was going to develop the same genetic conditions. Certainly.
Okay, thank you for your answer. And we will be taking now the last questions from the audience. So we have one more question.
First of all, thank you very much for your presentation. My question basically involves what do you think of the current situation of genetics, especially in education? Do you think that what's included or what's thought about that we learn about genetics, is it sufficient for the future, or do you think there should be some points added? Should it be sort of updated? Do you think that we are ready now, the knowledge that we have now for those who are already studying now, but those who finish, for example, in Greece or even in Europe? Do you think that it can be improved, or do students have to be a bit more informed? Yeah.
Very nice question. Let me speak again with facts. That's my favorite thing. If I ask you in the audience, adenine pairs with cytosine, yes or no? Then you probably hear, what does the guy say now? No, okay? 13.2% said yes in a study that we ran. That's actually published in 2014, Personalized Medicine. If I tell you that humans have 48 chromosomes, would you say yes or no? No. 33.2% said yes. Healthcare professionals, we're not just asking people on the road, healthcare professionals. So that means that we should be very persistent in keep up the genetic education of healthcare professionals, particularly the older generation. Okay? So it's an endless effort, I can tell you. And since we're in Greece, I can tell you what ancient Greeks were saying. They were saying that you can plant an olive tree knowing that you'll not be around to see it and eat olives from it. So you should be very persistent, you should try endlessly. And running again a similar study that was published in Public Health Genomics in 2014, we have seen in 11 southeastern European countries that the level of education of pharmacogenomics undergrad and postgraduate is immensely heterogeneous. There are some countries that they didn't taught pharmacogenomics at all, both levels, and other countries that are more advanced. Believe it or not, Greece was among these countries that were more advanced, having pharmacogenomics undergraduate and postgraduate level. And we're talking about 200 universities, more than 300 departments. So it contrasts the situation that is currently in the north or Western European countries. Again, it's an issue of harmonization. How long does it take? We don't know. We need to start working on that.
Thank you a lot for your answers. Are there any other questions? Last questions? Burning questions? I can't see any. Okay, so we will be finishing now the panel discussion. I would like to thank all our panelists for all your answers, explanatory answers. There were so many questions, and I'm sure we all learned so much, so thank you a lot. And now we will move on to the next point, so Student Symposium. So now I would like to give the floor to one of our college students, Elena Desiti. She will present her work. Thank you.
Hello everyone, my name is Elena Desiti and I'm a fourth-year finance student at the National Competition University of Athens here in Greece, and I'm a member of CPSF. Before we start, I would like to thank the organizing committee of the symposium, and I'm going to present you my bibliographic research on anatometrics and T-cell platforms linking skin cell functionality for skin regeneration that I performed under the supervision of my professor, Martina Black. So, firstly, we're going to see what nanocosmetics actually are and where they can be used. Nanocosmetics are actually cosmetics containing nanoparticles that are structured to size between 1 to 100 nanometers. And nanocosmetics, compared to conventional cosmetics, have more enhanced performance and brightening properties such as color, texture, transparency, solubility, and durability. Nanomaterials can be used in cosmetics as transdermal drug delivery systems by delivering the active compounds more effectively to target site. And nanomaterials have been used in many products, such as moisturizers, where better moisturization occurs due to the reduced particle size, and sunscreens, where you have more enhanced UV protection. Here you can see the examples. Therefore, the critical question is, do nanocosmetics actually have an impact on skin regeneration? And in order to answer this question, we have to see the mechanism of skin regeneration very deeply. As you can see in the picture, the skin has three layers: the epidermis, the dermis, and the hypodermis. And each layer of the skin can regenerate itself by different mechanisms. The external layer is the epidermis, which regenerates itself by the proliferation of keratinocytes and shaped available. Then we have the dermis, which regenerates itself by integration and proliferation of fibroblasts. And the deepest layer, the hypodermis, contains mostly water and fat, and also many hormones are produced there. So, as you saw, skin regeneration is really important in order to protect the human body from bacterial infections. That can be critical. And also, taking into account that many people deal with skin damages all over the world, it is very important to find more effective solutions to this worldwide problem. And nanocosmetics, and more specifically, artificial platforms mimicking skin cells functionality, are a new approach. And now I'm going to present you some of them. Okay, first we have a plasma-fluidic electrospan gelatin-based scaffold. As you can see in the picture, a scaffold is an extracellular matrix which provides a natural environment for the tissue to form. And this scaffold has wound-healing properties due to its ability to increase the pace of the cell's proliferation. Also, it has a more uniform attachment to the wound than the conventional cosmetics, and a more controlled drug delivery of many growth factors. Here is another scaffold, but this time it's a stem cell-based scaffold that can be used as a substrate for the differentiation of adipose-derived stem cells, or known as ADCs. These stem cells secrete growth factors and result in skin regeneration. And also, this scaffold contains collagen. Here is another scaffold, and it's a mimicking fiber scaffold, enriched with two growth factors: the VEGF, that stimulates skin healing by collagen deposition, and the VEFG, that accelerates fibroblast proliferation and has anti-scarring properties. In vivo experiments using a diabetic mouse model showed that this fiber scaffold is an effective alternative for diabetic wound healing, which is really remarkable. The last scaffold is a nano-complex scaffold based on surface modified PCL or polycapolacton nanofibers containing curcumin. Curcumin is actually a substance with skin regenerative properties that can improve the collagen deposition and reduce the wound healing time. Also, the scaffold contains PET methyl ether methicillate surface and PCL nanofibers, and this is why it is so effective on delivering the curcumin to the target site. Moving on to the next category, nanoparticles loaded with keratinocyte growth factor 2 are really effective on skin regeneration. The administration of those nanoparticles loaded with KZF2 compared to the free KZF2 a better acceleration on the cell's proliferation, reducing the wound healing time. Next, we have the silver nanoparticles, and these nanoparticles enhance the keratinocyte and fibroblast proliferation by suppressing the innate immune system, resulting in increasing the wound healing rate and decreasing the scarring process rate. It's a really remarkable case of toxic dermal necrosis, almost 70% of the patient's body, that was caused by carbamazepine administration. And exclusive treatment with silver nanoparticles was applied, and the wounds were covered with bandages. And after only 9 days of treatment, full recovery was observed. So you can see how effective nanotechnology is compared to the conventional therapies. And in the picture you can see different images of silver nanoparticles with different shapes. First we have the nanospheres, the nanogrins, the nanobars, the nanowires, nanocubes, pyramids, nanolars, and nanoflowers. Next we have the gold nanoparticles that not only have regenerative properties but also anti-inflammatory and antibacterial. These gold nanoparticles show the antibacterial spectrum towards gram-positive Staphylococcus and gram-negative Escherichia coli. Also, these gold nanoparticles slow down collagen degradation and elastin degradation. And in the picture you can see in a case study where those nanoparticles were applied in the mice model, how quickly the wounds were regenerated. The skin was regenerated. Next we have the zinc sulfide nanoparticles. In a recent study, it was investigated for wound healing on a rat business model, and it was discovered that those nanoparticles stimulated rat skin fibroblast proliferation, reduced the wound contraction, and enhanced the re-epithelialization. It was found that these nanoparticles can be a promising approach for wound healing. Next we have the rosy nanotubes, and those nanotubes belong to the category of nanohydrogels. And also these nanotubes have a similar nanostructure as keratin and collagen would naturally appear in the skin, so this is why they are so effective. And in an interesting study, showed that hydrogel impregnated with epigallocandin is very effective in killing wounds by improving angiogenesis and restoring epithelialization. And lastly, we have the method relief and-dry modified gelatin hydrogel and mimic neutrophilic nanoparticles hydrogel. This hydrogel was originally created for diabetic wound healing, and in the picture you can see what this wound is, the diabetic wound. In the recent study has shown that this hydrogel might be a potential treatment for diabetic wound. The research was conducted in vivo on type 1 diabetic rats, and the diabetic wound rats that received the hydrogel almost fully healed with no formation of scar after 21 days of treatment. And you can see how quickly this happened, because it's really important to see the difference between the nanohydrogel compared to a conventional gel that it is usually applied on the wound. So now it's time for a brief summary of what we learned today. As we all saw, nanocosmetics and nanotechnology in general enhances the properties and helps to deliver more effectively to the target site the active compounds and increase the biocompatibility and the bioavailability. Therefore, it is really difficult to choose which nanoparticle or nanocarrier to choose every time. So it's why it's important to do more biological tests and more research to have the most effective and safe product. So nanocosmetics and more specifically those artificial platforms mimicking skin cells functionality seem to be really effective, but of course more research is of paramount importance. Here's the bibliography, and thank you for your attention.
Thank you, Stella, for your presentation. Are there any questions? Thank you a lot. Stella's presentation was the last point for today's symposium. Yes, with this being said, I would like to thank you to all of our speakers for sharing their knowledge. And to the participants for your active involvement in all the questions. It was great to hear your questions and interest in the topics presented. And now I would like to inform you that we will have a lunch break. Please keep in mind to be on time for workshops at 2:30. Also, I would like to remind you that on participants group, you got the link to feedback form, so feel free to also fill in the feedback form for our sessions, for symposia and workshops. And I would like to remind you also that on Friday and Thursday, you have to attend all sessions, all workshops in order to receive the certificate of attendance. So this is our last symposium session. I would like to thank you once again for your attention. And I wish you a wonderful rest of the Congress.